All right, let's dive right in. Today, we're basically cracking open the book of life itself, and we're going to show you exactly how scientists learned to rewrite the text inside. So, let's just start with a huge question, one that honestly sounds like it's straight out of a sci-fi movie, but it's happening in labs as we speak. What if you could edit the very code that makes a firefly glow, or, say, makes a plant totally resistant to disease? Well, believe it or not, way back in the 1970s, that sci-fi question, it became reality. A couple of scientists figured out how to do it. They discovered biology's cut and paste tools. And no, we're not talking about a Word document here. We're talking about the DNA that makes you, you. This discovery, it's literally the foundation of all modern biotechnology. So let's get into it. What is biotechnology really? I mean, the word itself sounds super futuristic, right? But its roots, they actually go way, way back. Still, what we're talking about today is, well, it's a whole different ballgame from those ancient practices. Yeah, I mean, if you want the official definition, the European Federation of Biotechnology has a good one. But think about it like this. For literally ages, humans have been using living things to make stuff. Beer, bread, wine. That's biotechnology. But modern biotechnology, whoa, that's like going from a horse and buggy to a warp-drive spaceship. It's all about diving deep, right down to the molecular level into the DNA itself, and engineering it to do completely new things and do them on an industrial scale. Okay, so how did this whole revolution kick off? Well, the story really gets going with a chance meeting. A couple of scientists whose research, it turns out, fit together absolutely perfectly. I mean, like two sides of a zipper. So check this out. On one side, you've got Herbert Boyer. He's at the University of California, and he's studying these enzymes from E. coli bacteria. And what he figures out is that they act like, well, like tiny, super precise scissors that can cut DNA strands. Then on the other side, you've got Stanley Cohen over at Stanford. He's the expert on these little rings of DNA called plasmids. And his whole thing was figuring out how to pop them out of bacteria and then stick them back in. So you see where this is going, right? One guy has the scissors, and the other guy has the delivery truck. And then, boom, the breakthrough. It's 1972. They're at a conference in Hawaii, of all places, and they start talking, and they realize, hey, let's put our two ideas together. So they do. They take a gene for antibiotic resistance, snip it out using Boyer's scissors, and then they paste it right into one of Cohen's plasmids. That tiny little loop of DNA they created, that was the world's first piece of man-made recombinant DNA. And just like that, everything, and I mean everything, changed. So what this breakthrough really did was give scientists a brand new toolkit. I'm talking about a set of molecular instruments that lets them perform, basically, genetic surgery with just incredible precision. Let's open up this toolkit and see what's inside. Okay, tool number 1, restriction enzymes. This was Boyer's big thing. You got to think of these as the molecular scissors. They scan along a strand of DNA, reading the code, and when they find their exact target sequence, let's say GAATTC, chop, they make a cut. And here's the really cool part. The cut isn't always clean. It often leaves this little overhang, a sticky end that's just dying to pair up with a matching piece of DNA. And get this, we know of over 900 of these things now, so we can cut DNA with mind-boggling precision. All right, so you've made your cut. Now what? You need a way to move that little piece of DNA into a new cell, right? Well, that's where Stanley Cohen's plasmids come into play. They act as vectors, or to put it another way, they're like a biological USB drive. You can load your gene onto it and then plug it right into a host bacterium. And last but not least, you've got to stick it all together. You've got your gene, you've got your open plasmid, you need some glue. That's a job for an enzyme called DNA ligase. It's basically molecular superglue. It seals the deal, joining the two pieces of DNA together permanently. And voila, you've created your brand new custom piece of recombinant DNA. Okay, so let's recap the toolkit. We've got our scissors, restriction enzymes, we've got our USB drive, plasmids, and we've got our glue, DNA ligase. So now let's see how scientists actually use all this stuff, step by step, like a little assembly line for building with DNA. And it really is a surprisingly straightforward process when you break it down. First, you got to isolate the DNA you want to work with. Then you use your enzyme scissors to cut it up. Next, you glue the specific piece you want into your plasmid vector. Then comes the tricky part. You transform a host cell, which just means you get that new DNA inside of it. And finally, you amplify it. You just grow massive batches of these newly modified cells. So yeah, step one is all about getting the raw material. To do that, scientists have to break open cells, usually with some other enzymes, to get to the juicy DNA inside. And then, here's the super cool part. They add some ice-cold ethanol to the mix, and the pure DNA actually separates out from the liquid. It looks like these fine, wispy threads. You can literally see the code of life with your own two eyes. Okay, but how do you know if your scissors actually worked? I mean, you can't see the cuts. Well, this is where it gets really interesting. There's this technique called gel electrophoresis. You put your chopped up DNA into a gel and run an electric current through it. Since DNA is negatively charged, it gets pulled through the gel. And just like running through a thick forest, the smaller pieces can move faster and farther, so it separates everything by size. Then you add a special stain, flick on a UV light, and bam! You see these bright, glowing orange bands. Each band is a different sized group of DNA fragments. But wait a second. What if you only have a tiny bit of DNA to start with? Like a single molecule from a drop of blood at a crime scene. You can't work with that. You need more. Well, that's where another game-changing technique called PCR comes in. PCR stands for polymerase chain reaction, but all you need to remember is that it's a genetic photocopier. It can take one single piece of DNA and make millions, even billions of copies in just a few hours. It's absolutely incredible. Okay, so you've made billions of copies of your custom DNA. Awesome. Now for the next hurdle. You've got to get it inside a living bacterium. And these little guys have pretty tough cell walls, kind of like a security gate. You can't just knock and ask to be let in. So what scientists do is make the cells competent. It's a fancy word that just means they make the cell walls leaky for a second. They often use a method called heat shock, a little bit of calcium, a quick jolt of heat, and for a split second, tiny pores open up, just long enough for that new plasmid to sneak inside. And once you've got that one single bacterium successfully transformed with your new gene, well, the real magic begins. The final stage is all about scale. It's about taking that one cell and turning into billions, even trillions of identical copies, all of them little factories carrying the exact same instructions to make whatever you want. I mean, just look at the incredible leap in scale here. We started with this tiny microscopic strand of DNA in a test tube, and we end up with these massive industrial bioreactors. That's the real power of biotechnology right there, turning a tiny little piece of genetic code into a full-blown factory that can churn out tons of product. And don't think of these bioreactors as just giant mixing bowls. Oh no, they're more like 5-star, all-inclusive resorts for our engineered bacteria. Everything inside is perfectly controlled. The temperature, the pH level, the nutrient food supply, the oxygen levels. Everything is optimized to make those trillions of tiny cellular factories as happy and productive as possible. Also, they keep churning out that one specific protein we need. And at the end of the day, that's what this is all about. This entire mind-blowing process, it almost always boils down to one single goal: producing a desirable protein. And that protein could be anything. It could be life-saving insulin for people with diabetes. It could be a cutting-edge vaccine. It could even be the enzymes in your laundry detergent that get out those tough stains. It's all about taking a tiny snippet of code and turning it into a real-world product that can genuinely change our lives. But hold on, we're not quite done. When the bioreactor switches off, there's one last absolutely critical step. It's called downstream processing. This is the cleanup phase. You have to separate your precious protein from all that other gunk, the trillions of used-up cells, the leftover nutrient soup. Then you purify it to an incredibly high standard, mix it with preservatives to keep it stable, and finally, package it all up so it's ready for the market. So there you have it, from a casual chat at a conference in Hawaii to a massive, world-changing global industry. We now have the tools to edit the source code of life itself. We've pretty much figured out the how. The much bigger question, the one we all have to think about now is, what should we write?